Okay, let's move into Unit 2. So Unit 2 deals with soil-plant relationships, and this is a relatively important unit. Every unit's probably important, but this one's relatively important because we'll describe basically how plants see and absorb and take up nutrients. And so there are 6 learning objectives. The first objective is to describe the differences in taproot versus fibrous root systems and know roughly what types of plants have these two different types of systems. For example, the differences between legumes, which typically have tap roots, and grasses, which have fibrous root systems. And we'll talk about that briefly. Objective number 2, understand the relative importance of root hairs and lateral roots in nutrient absorption and know where root hairs develop within the rooting system itself. Objective 3, you should be able to list the functions of roots. There are 4, and describe where they take place within the root system. Objective 4 is describe the mechanisms of root interception, mass flow, and diffusion, and how they supply nutrients to plant roots. Each one is slightly different. And then tied together with objective number 4 is objective number 5. And number 5 is that you should be able to identify the primary mechanism responsible for nutrient transport of nitrogen, phosphorus, potassium, calcium, and magnesium with respect to root interception, mass flow, or diffusion. So we'll tie both 4 and 5 together. And then objective number 6 is to describe active transport and its role in nutrient uptake from soil to plants. So mass flow, diffusion, and root interception are considered passive uptake mechanisms. Plants, yeah, they have to extend a little bit of energy to do this, but not as much energy as compared to active transport. So objective number 6, we'll talk about active transport. So let's do this first. Let's focus on objectives 1 and 2. So describe the differences between taproot and fibrous root systems, and then understand the importance of root hairs and lateral roots in terms of nutrient absorption. So there's 2 types of roots. There's tap roots and there's fibrous roots. And it's always rather difficult to get a picture of what roots look like because they're below ground, and to pull them out of the ground, you destructively sample. But the picture in the top right-hand corner did a pretty good job of tagging with this green dye what corn plant roots look like. And hopefully most of you understand that corn is a grass, and in fact, most grasses have a fibrous root system like you see in this picture in the top right-hand corner. This is considered fibrous. And grasses tend to look like this. So here's basically wheat, and that's a fibrous root system, and this is sugar beet, which has a taproot. The whole thing is a taproot. And then you have other types of plants, and these, of course, are hand drawings, but this is called blazing star. It's a prairie plant, which has a long taproot, and it can go down meters, versus this is called wiregrass. It's another prairie plant, and it's a grass, and it's fibrous root system. And so each one of these lines in this picture on the left-hand side represents 1 foot. So what's it? 1, 2, 3, 4, 5, 10, 15, almost maybe 20 feet deep. And that's not uncommon for most taproot systems, unlike sugar beet. But regardless, these plants are adapted to explore the soil in different ways. And these taproot systems explore deeper in the profile, mainly for obtaining water. And these fibrous root systems are here for not only obtaining water, but also looking for nutrients. And they they both act in different ways in terms of, oh my gosh, if you want to talk about carbon storage in systems and offsetting CO2 emissions in the atmosphere, if you could somehow genetically modify plants to increase the amount of carbon that's being pumped down deeper into the soil profile over time, you likely will see an increase in organic carbon contents deeper in the profile, because this is what happens in soil profiles, is the deeper you go, the more anoxic it becomes, the less oxygen can diffuse down deeper into the profile. And thus, microorganisms that help decay organic materials don't go very deep as well, because most of these are most of them are aerobic organisms, so they need oxygen to survive. And so scientists are actually doing this. They are taking a look at taking specific genes out of one crop that can pump carbon down deeper into the profile, and potentially putting them into other crops. It's actually a rather fascinating field of study if you're ever interested in doing something like that with respect to global climate change and carbon sequestration in soils. Regardless, you get the idea. There's taproots and there's fibrous root systems. So the root structure itself, what do we typically see when we look at roots? We find that the root is not just one tube, it's tubes or a root with lateral roots coming off of the sides of the main root. And so there's several things that occur when roots develop and grow. And I like to start by talking about the root cap itself. So if you're looking at the root cap, it's this area right at the tip of the root. And these are cells that basically slough off they develop, and they slough off as the root grows. And what that root cap does is it protects the apical meristem. The apical meristem is the growing tip of the root itself. And so this root cap aids in root penetration through the soil, and it actually plays a role in the plant's response to gravity, which is highly important. So the plant knows which way to send the roots and which way to send the roots. This root cap, or what you see here in this picture here, it's a portion of the root. It shows the relationship between the root cap, the region of root hairs, which typically are derived near the tip of the root. Okay, and then they slough off and they die, and the root becomes thicker, and lateral roots emerge, which contains the root cap and root hairs that grow and die quickly. Okay. The lateral roots actually emerge from within the 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 vascular bundle or vascular tissue itself, and we're going to talk about that here in a second. So this is what occurs. All right, so you have something called the primary meristem, and the primary meristem is the young root that has yet to be developed or fully developed. And so what you see here are a number of different types of tissues. And this is primary meristem from onion root. And there's several items or areas that are developing, that develop into the root itself. Okay, so the primary meristem is what you see here in this picture, and it consists of the protoderm, which actually ends up being the epidermis, or the outside of the root hair or the root itself, the ground meristem, which ends up being storage, and the procambium, which ends up being the vascular tissues for conduction of carbohydrates and nutrients. And then, of course, you have the root cap here, and you can see the apical meristem. See this curved section of cells? Oops. This curved section of cells here is the apical meristem. That is the actual growing tip of the root. And so let's talk about these. So you should have a good idea of the underlying terms on this slide. So I mentioned this on the previous slide. The protoderm, when it grows and becomes the actual root, it becomes the epidermis. And the epidermis, or the epidermal cells, turn into, on occasion, root hairs, which are used for absorbing water and nutrients. This occurs in epidermal cells. And the protoderm is the precursor to epidermis cells. The ground meristem, sort of this muddy green portion here, becomes something known as the cortex. And the cortex in roots is where starches and other compounds are stored. They're actually specialized cortical cells that also form the epidermis. All right, so the epidermis comes from basically two regions, but I like to think of the glomerulus as becoming the cortex. So it's a storage facility for carbohydrates, i.e., energy in plants. And then the procambium, right in the center, becomes the vascular tissue. Also, maybe you've known it as the xylem and phloem. And this is used for conduction of transport of water and nutrients up into the plant and carbohydrates down to the roots. So here's a picture. This is a transverse view of a willow root. And this is an overall view of the root nearing completion of primary growth. So you can see, you don't see the procambium anymore, and you don't see the other precursor features. What you see here is the epidermis that surrounds the entire root, right? Looks highly structured. And then you've got this cortex, which looks, oh, like a mosaic, but this is for storage of carbohydrates and other compounds. And then you have the vascular bundle in the center that's comprised of the xylem and the phloem. Hopefully you've seen some of this before in your other coursework. And so let's talk about root hairs. And when we talk about root hairs, and oftentimes when we talk about compounds or materials or substances in soil that are really small, we talk about the importance of surface area. And that is the case for root hairs. Root hairs are relatively small, they're abundant, they're near the growing tip of the root itself. And because they're abundant and they're very small, they have a large surface area for absorption of nutrients and water. And we're going to come back to this theme of surface area again in one of the subsequent units. So this is a picture of the apical meristem, likely the root cap of bent grass. The root hairs are what you see that look like silhouettes coming off the root itself. These, it says, can be as much as 1.3 centimeters long, and they can attain their full size within hours, which is unbelievable. All right. The root hairs are there to absorb water and nutrients. They're short-lived, but there's formation of new root hairs as this root tip grows down through the soil. All right. And so these root hairs will eventually fall off, they'll die, they'll add carbon back to the soil, an energy source for microorganisms. So there tends to be a lot of microorganisms living near where these root hairs are or near the tip of the root itself. And these root hairs, like I said, they're very small, they grow quickly, and they have a lot of surface area for water absorption and nutrient absorption. So where do these root hairs come from? These root hairs, it's not magic. They actually come from epidermal cells, right? So here's a picture of corn with root hairs emerging from the epidermis, from the very outer edge of cells, all right? The epidermis is where the root hairs develop, and they come from specialized cells in the epidermis called trichoblasts. And I'm not expecting you to remember that term trichoblast, but you'll notice that not every single epidermal cell is turned into a root hair, but some are triggered, and they're triggered to create or generate root hairs to explore the soil for water and nutrients. And they come from the epidermis. So you know that when you look at a root, when you rip a plant out of the ground and you look at the root is not just one tube or one root going down. There's lateral roots, right? And you've seen that in the pictures for the fibrous root systems and the taproot system. And so where do those other roots come from? Well, those other roots, or the roots that are lateral to the main growing point of the original root itself, those roots are called lateral roots. And lateral roots emerge from the vascular cylinder or the vascular bundle. And then they send out root tips, right? So there's a root cap, there's a apical meristem, and then there's root hairs for exploration of water and nutrients. And this continues over and over and over until the plant dies, right? So this is a picture of a vascular cylinder where the lateral root is emerging from corn, and you can see it emerging here. And I would imagine that hurts a little bit. If I was a plant, I'd be like, ooh, that probably hurts a little bit. Regardless, it comes from the vascular cylinder. And that should make perfect sense. And it's amazing that it does this because this lateral root needs to be connected to the vascular cylinder in order for nutrients and water that are obtained from this lateral root to be pumped up in the plant. So here are some examples, and this is from Willow again. And this is the vascular cylinder in the middle, and you see this purple bulge here. That's the beginning of a lateral root. And here's another picture that shows two lateral roots emerging from the vascular cylinder. And then this third picture shows one lateral root that's erupted out of the epidermis and another one about to emerge from the vascular cylinder of this willow. And it's amazing that this even works because when this lateral root emerges, it has to disrupt the outer membrane of the root, but the contents don't leak out, right? So there must be some connectivity between the lateral root and the existing epidermal cells. Regardless, this is how roots grow. So, based on what I just taught you, which type of root structure contributes the most to nutrient uptake? Root hairs or lateral roots? And you should all be thinking root hairs because root hairs are small and they have a lot of surface area. And so they see a lot of soil, relatively speaking, as compared to lateral roots. And that's where the magic happens in soils in terms of water absorption and nutrient absorption. It's through the root hairs. Okay, so let's stop here and we'll pick up with the next objective in the next video.